Hey everyone, we're going to pick it back up with the digestive system, and we are talking about the large intestine. So the large intestine looks something like this. There is a boundary between the final end of the small intestine, which is called the ileum, and then this first pouch into the large intestine is called the cecum. And there's a valve here actually, which is going to prevent things from the colon from getting back into the small intestine, and this is important. There's a lot of bacteria that are inside of the large intestine, and the small intestine is mostly sterile. There will be bacteria there, but not nearly to the degree that they're found in the large intestine. And there's about 700 different species of bacteria in your large intestine, and on a cell-to-cell count, I've heard that there are 10 times more bacteria in your gut than there are of your own cells. So you're 10 times more bacteria than you are you. Now, bacteria are much smaller than eukaryotic cells, so that doesn't present as being a large volume to your body. But in either case, a lot of bacteria here. Now, before I get into the portions of the large intestine here, I just want to point out that the bacteria found in your colon are helpful to you. They are going to be a wonderful display of symbiosis, because what they do is they take all of the more complicated lipids and proteins, and they help to break them down even further. So they're assisting the digestion. They are also creating vitamins for you. There are several vitamins that you can't make. Generally, vitamin K and B12 are the ones that are talked about. And these are not ones that you can make yourself, but the bacteria make these vitamins, and then you absorb them. Furthermore, if the bacteria in here get disrupted, it can lead to a lot of problems. For example, there's a clinical application here of Clostridium difficile, which is a type of bacterium that can invade your gut if all of the microbiome, all of the bacteria that live there, are destroyed with antibiotics. So if somebody's just had a surgery and they've treated with antibiotics, the antibiotics will kill all the bacteria that are in this region. And then this particular strain of bacterium is very quick to seed the area and flourish. I like to think about it like if something traumatic happens to your yard and all your grass dies, then the weeds sprout up because there's bare soil, and then they spread like crazy. Well, that's sort of what's happening here. And these create a toxin which makes people really sick. I've heard of people passing out, having low blood pressure, feeling like they're dying from all of the effects of that toxin. So, interesting situation there. And one of the workarounds would be to seed the colon again with a healthy array of bacteria. So they'll actually do what's called a stool transplant, and put a colonoscopy scope up here, and or something up here, and they'll inject a slurry of donated fecal matter into the colon to help seed the bacteria back into the colon. All right, well, that's the bacteria, and the anatomy, or the anatomical layout of the large intestine, looks something like this. You have the ascending colon. This is on the right side of your body, so I always just remember you have the right to ascend. It helps me remember that the ascending colon is on the right side. Then there's going to be a flexure, or a bend, and then there's going to be the transverse colon. That's the one that goes from left to right. Then there's going to be another flexure, another bend, and then there's the descending colon, and then lastly the sigmoid colon here. Okay, once we make the S curve, sigma is the Greek letter for S. So after we make the S curve, then we run into the rectum, and the rectum will be the final place that fecal matter is stored, and then from there it's defecated or expelled out of the anal canal. And some interesting mechanisms there that we'll take a look at a little later. Okay, some other features that we could see are the haustra, which are these little pouches. And the haustra, I like to think of them almost being like an accordion type structure. There's ribbons of muscle. There's 3 ribbons of muscle that run circumferentially around this, or longitudinally rather, along the colon. And those ribbons of muscle, you can see them somewhat here. Those ribbons of muscle are called the tenia coli. And what they do is when they contract, they scrunch the colon down a little bit to create the bulges of the haustra, and then when they relax, the haustra sort of dissipate and the colon recoils. So that's happening quite frequently. About once every 30 minutes or so, you'll have the contraction of the tenia coli, and you'll have the haustra bulging out like that. And it just helps to churn and mix the contents in the bowel. Another thing that can happen is what's known as a mass movement, and this happens 3 or 4 hours a day, or 3 or 4 times a day. This is what happens just prior to you feeling like you need to go to the bathroom. The whole colon can squeeze down and very quickly push all the stool material towards the distal end of the colon, and then shortly thereafter you'd have the urge to defecate. So some interesting motility features there. Other things about the colon, I'm trying to think here. Talked about the bacteria, we talked about the haustra and the tenia coli, and this, yeah, I think that's good for now. Okay, now let's talk about the defecation reflex. So this is what happens when you feel like you have to go to the bathroom. What's an interesting layout is that there's 2 rings of muscle. There's the internal anal sphincter and there's the external anal sphincter. And the internal sphincter is under involuntary control, meaning you can't contract it or not. It just will relax on its own. The outer sphincter is in voluntary control, meaning you actually can change that that contraction. So how it works is as the rectum becomes distended, there's a stretch reflex here. There's stretch receptors that are going to feed back and cause a relaxation of the internal sphincter. Okay, this is the moment where you go, oh, I gotta go to the bathroom, because as soon as this relaxes, the only thing keeping that stool from passing out is you consciously contracting the external anal sphincter. So you'll feel that. You feel this is like the prairie dog in it type situation. Well, maybe not quite that bad, but this is where you're actively making sure that the stool doesn't come out, and then once you get to the toilet and you're comfortable, you can relax that sphincter, and then the stool will pass out. Okay, that's going to bring us then all the way down through the alimentary canal. I did just want to talk a little bit about some of the hormones that help control digestion. Now that we've talked through all the anatomy of the alimentary canal, we've mentioned gastrin already. This is going to increase the stomach acid and the motility. And some of the things that are going to cause this release would be distension of the stomach, if there's an elevated pH in the stomach, or if there's undigested proteins that are found in the duodenum. Okay, another couple of hormones are these. There's a hormone known as secretin, there's gastroinhibitory peptide, there's cholecystokinin, CCK, and there's vasoactive intestinal peptide, the VIP. All these hormones are going to be secreted when there's chyme that enters into the duodenum. Okay, so this duodenum here gets extended, these hormones are released. If that chyme has a really low pH, it would tell the, it would say that there's been some adequate digestion occurring, because gastrin was likely released prior, increased the amount of acid, which decreased the pH. So if there's a low pH content, that can be detected by chemoreceptors and also used to stimulate the secretion of these hormones. Okay, if the chyme has digested proteins or lipids or carbs, these will be released. And some of the roles of these hormones are the following. Secretin is going to increase our bile production in the hepatocytes of the liver. So it would actually dissolve into the blood, feed back all the way to the liver, and cause the hepatocytes to start releasing more bile. Okay, it's also going to be targeted. There's going to be a target on the pancreas for it, and that's going to cause more bicarbonate ion to be made, or buffer secretion to be made. GIP, this is going to actually pump the brakes on gastrin a little bit. It's going to inhibit, as the name suggests, gastrin inhibitory peptide. It's also going to act on the pancreas to stimulate some insulin production, which will be useful in helping us absorb the digested carbohydrates, principally glucose. Okay, CCK is going to increase the production of pancreatic enzymes, and it also opens up the hepatopancreatic sphincter. So that was, again, this little opening here, which was a merge point between the accessory organs of digestion, the main pancreatic duct here coming in here, and then the common bile duct coming in here. There's a sphincter there, and in the presence of CCK will help to relax that, so bile and digestive enzymes coming in from the pancreas can be injected into the duodenum. Similarly, CCK finds its way to the gallbladder and is going to cause contraction of the muscle of that gallbladder, and that's going to help to squirt more bile into the duodenum. This is a little schematic here showing how this is illustrating, or how this all plays out. Bile can flow passively through here, but generally if it's accumulating, it will get concentrated in the gallbladder, and the more that it stays there, the more concentrated it becomes. Okay, and then what CCK will do is it will release the muscles that are closing that sphincter, and it will also cause contraction of the muscles in the gallbladder, both of which will push bile out. And bile is a big emulsifier, so what it does is it takes the big fat droplet and it breaks it into little droplets for you. All right, vasoactive intestinal peptide, then it dilates your capillaries in the digestive organs. It increases blood flow to your digestive organs so that there can be more absorption of all that has been digested in the stomach. Okay, a little bit more of a story here on the chemical digestion then and absorption. What happens specifically for each of the different macromolecules. For carbohydrates, it looks something like this. The largest carbohydrates would be something like a polysaccharide like glycogen, and that is acted on by salivary amylase. Okay, you release this in your saliva, and it's going to start working right away on the glycogen to break it down into smaller bits. Eventually, it could get all the way down to the disaccharides. And there's different kinds of disaccharides. There's maltose, there's sucrose, and there's lactose. Sucrose would be glucose and fructose. Lactose would be glucose and galactose, and maltose is two glucose molecules put together. And each of these then have their own enzyme that works on it. Okay, so the disaccharides, maltose gets acted upon by maltase, and then it makes the individual monomers, which are two molecules of glucose. In this situation, sucrose is worked on by sucrase, and then it yields glucose and fructose. And then similarly, lactase converts lactose into glucose and galactose. These are all brush border enzymes. So while there might be some salivary amylase and also probably maybe some pancreatic amylase working to create it into the smaller bits, it's only at the brush border, which if you remember was the microvilli, these little folds on the membrane of columnar cells. It was the microvilli that would make these enzymes, and then only then when the disaccharides brush across the brush border would the enzymes work to bring them down to the individual monosaccharides. And then these are what are eventually absorbed into the cell. Okay, for proteins, it looks something like this. You have a larger protein. Pepsin starts working away in the stomach, and then that's going to break down the proteins into larger polypeptides. And then here we have some pancreatic enzymes that are going to be used to convert or break down larger polypeptides into really short peptides or even individual amino acids. The pancreatic enzymes that do this are chymotrypsin, trypsin, and from here to get down even the furthest way possible all into individual amino acids, this would be the brush border enzymes again. And there's two more notable ones. There's aminopeptidase and dipeptidase. Okay. And then from here, we can absorb it across the simple columnar epithelium located in the small intestine. Now, how the body will absorb these different molecules varies, and there's some different cellular mechanisms at play here. We won't go into a huge amount of detail here because this isn't really a cellular biology class, but generally speaking, I think it's helpful to know some of the physiology of the transport mechanisms. So here's a table in your book, table 2310. And to illustrate a little bit about how some of these mechanisms work, I pulled a figure from a publication that looks like this. So this is illustrating specifically glucose and fructose, but it's going to be somewhat of a similar story for a lot of the, for some of the others here. So what we have, this is showing an epithelial cell, so this would be like the simple columnar epithelium inside the small intestine. And here would be the apical side of that epithelium, and then this would be the basal side. Well, there's a whole bunch of things that are transported with what are known as co-transports, and a co-transport is something that usually flows down its concentration gradient, and the flow of something down its concentration gradient actually then fuels the energy to push something up its concentration gradient. So in this scenario, you actually have a lower concentration here for glucose and a higher concentration inside the cell. And so glucose is being pushed up its concentration gradient. That always requires energy to push something up its concentration gradient. And in this instance, the energy is provided by the flow of sodium going down its concentration gradient. There's a lot more sodium outside of a cell than there is inside of a cell. And so it's a clever way to get something to go up its concentration gradient. Well, this is called the sodium glucose cotransporter, or SGLT1 is the name. And on the flip side, there are some carrier proteins here which are not transports. They don't use secondary active transport. They would use facilitative diffusion. So in this instance, fructose is not generally held in the cell to any high abundance or concentration. It gets converted to other things. So there's generally a lower concentration of fructose hanging around and a higher concentration outside of the cell. And the fructose will flow through the carrier protein through facilitative diffusion. It's going down its concentration gradient. And then from here, it will just continue to flow out, going down another step. Okay, so you're going from high to like medium or low to very low concentration. And then from here, it's now in the portal blood, so it can make its way over to the liver for processing. Now, the glucose also has a carrier protein at this point. So catch this, there's the sodium cotransporter to push it into the cell, but now once it's inside the cell in high abundance, it can diffuse down its concentration gradient. And it does that through this carrier, which is the GLUT2, the GLUT2. So you got GLUT5 and GLUT2 both being facilitative diffusion carrier transports. All right, then the only thing that's going to be required here is that all the sodium that's gone in, we have to pull it out of the cell to maintain a low sodium concentration here, because this whole mechanism hinges on the fact that we have a low sodium concentration. If this sodium starts to accumulate, well, now our gradient dissipates. We want there to be a high amount of sodium outside and a low amount of sodium inside, and that the steepness of that gradient will drive sodium in. If sodium accumulates here, then we lose our gradient and it's no longer going to flow, and then you can't get sugar inside of your body, which is a bad thing. Okay, so lucky for you, there is a sodium-potassium pump that is going to take that sodium, pump it out of the cell, and in exchange pump potassium in the cell. And that's going to continually remove away the sodium so that the gradient can be maintained. All right, now galactose is going to use a similar mechanism seen here. There's going to be a sodium glucose or a sodium cotransport That's also true of most of the amino acids. They are going to have a similar mechanism to this, where sodium flows in and it carries in the thing. All right, now looking at the lipids, the bigger lipids like a long chain fatty acid or a monoacyl glyceride, this would be a glycerol molecule that just has one fatty acid on it. These diffuse into the intestinal cells, and then from there they are packaged up with proteins, and then this is called a chylomicron. These are what form cholesterol, and you can have higher density ones that have a higher percentage of protein to the lipid content, or you can have low density ones which are more lipid rich. But they have these little protein scaffold and chaperone molecules that help package the fat and ship it around the body. Now, if something is really small, like a short chain fatty acid or the individual molecule glycerol, by the way, just to recall here from general biology, remember that triglycerides are a main way that fat is stored in your body. And that is going to be composed of one molecule of glycerol. Glycerol with then 3 fatty acids attached to it. So it's a triglyceride. Well, if we cleave these individual things off or these individual things off, then those can just diffuse into the cell through simple diffusion. There isn't any packaging of proteins necessary to make a chylomicron. They would just diffuse in, and then they would keep diffusing out. And then generally these will get absorbed by the lacteals of the villi, and then those will be brought through the lymphatic system back into the ducts that drain by your internal jugular vein on either side. And then nucleic acids, these have an active transport mechanism. So basically there would be a thing that they bind to, and then it changes shape and then pumps it in. Okay, so that's the story. We've got some sodium cotransporters for glucose, galactose, and amino acids. Fructose has got its own little thing. And then the bigger lipids get packaged into these chylomicrons, and they can be either high density or low density. The smaller lipids diffuse right in. Here's a picture, and just another picture showing the story here with lipids. You've got the big fatty droplets. They get emulsified into the small droplets. A really small fat droplet is called a micelle, just like almost like a pinpoint. It's really, really small. And then from here, they diffuse into the cell. They will get shipped out the back side, the basolateral side, and then they'll diffuse into a lacteal and be carried away from the intestine over to the blood. Or sorry, in the lymphatic system into the blood, and then at some point they'll go somewhere. I don't actually know what happens to these once they get into the lymph. If they play some role in maintaining the structure or the integrity or the composition of the lymph itself, I'm not entirely sure. It's sort of an interesting, curious mechanism there. Okay, that's going to wrap us up for the digestive system.